So I'm Brian. Heather is helping me today. Heather is our content lead and our kind of customer support. We're a small company, so we all wear lots of different hats. I'm a professor.
I'm a high school teacher, Brian's University.
Yes. So we combined, we know the pain of each different kinds of teaching. So
Sixth grade is a special kind, though. I taught 7th and 8th grade. 6th grade is something else.
Okay. So yeah, I'm Brian, and I'm a professor at BYU, Brigham Young University, and the chemistry department. And so we created these labs over about 2 decades, starting in 1998, and they've been through a lot of different iterations, but the principles behind them have been the same. And we developed it at BYU with BYU students, and using resources at BYU. They were sold for many years by Pearson, and several years ago we pulled back the licenses, and we've gone on our own as Beyond Labs. And so we've gone through and updated the labs and refreshed them and found a way to manage licenses and deliver them in a way that's accessible to lots of students through Mac and Windows platforms. So that's kind of who we are. So the labs have been used a lot for over the years. So we have lots of data, lots of use cases and how they're used, and what they're good for. And so feel free to ask questions, and we'll do our best to answer them. If you want to ask a question on the chat, Heather will answer if she can, and if she can't, she'll interrupt me, and we'll go that way. So we have a really wide mix. So I'm probably going to kind of show at a high level general chemistry, organic chemistry, and try to kind of hit all 4 kind of today in a high level, just to show what their coverage is. But once you kind of see how one of our labs works, they all kind of work the same. I like to say they're kind of like children. They're all children of the same family. They're all related to each other, but they're all different in their own ways. So so that's kind of how it works. So I'm going to share my screen, and so I'm going to tuck this out of the way down here. So let's talk about how we deliver the labs first. So what we made was we created a client, what we call a Beyond Labs client, that wraps and packages the labs, the worksheets, and the licenses all together in a simple package. So no more CDs if you were used to the old versions. Everything that's accessible as a download. So the client works on Windows 10, and Windows 8. We can get it to work, Macintosh, and so the way it kind of works is we have these different tabs. So these are the different labs. Mine are all installed, obviously, because I show them to people. But what you would see here is a little download button. So if you wanted to do chemistry, you would click there. So it would download, install it, and then you would access the lab through the client by clicking here, which I'll show you later. So we have different levels: high school, higher ed. The lab itself doesn't change at all, but what changes is the worksheets that you use with them. So that's how we adjust for level. So in the worksheet tab, this is where we find all of the lab activities that we've produced. By no means are these lab activities everything that the lab will do. It's a small fraction of the different experiments you can do in the lab, but these are meant to be the basic, most common experiments and activities that you would want to do in a classroom, high school, or higher ed setting. So the way it works is you would click on chemistry, then say, hey, I'm teaching higher ed general chemistry. I'd click here, and then they're organized in different topics. So if I wanted to go, say I wanted to go do atomic theory, then what I would do is click here on, say, Rutherford, and this would be a PDF file that you can save and download. Lots of different ways that you can deliver this to the student, but this is the simple way that we have, that we give the content, the lab activities. And so a little bit of background, but what's important here is that we provide step-by-step instructions on what to do and how to do it to perform certain experiments. So these lab activities become a really nice way to learn how to use the lab, how to use all the different interfaces. It's a way of doing it. So atomic theory, that may be something that no one really is a lab replacement because you're not doing Rutherford's experiment in a lab, but we also have things like acid base, so you're doing titrations. So we have almost 300 lab activities for all the different products. And so high school, we organize them by NGSS standards. And so they're easy to get to. So I'll show you how those work in a minute with the labs, but this is where the content is. Here's the license. So when you install the client, it comes with the 2-day free license for you to explore and play around with it. And then students can purchase a code directly, enter it into the activation box and submit it. And it gives you a license for a year, or schools can purchase a single code that we activate for a certain number of uses, and you hand out that code to students and they put it in and activate to get a license and to validate it. And finally, here in this part, we have the settings. Don't click this red button. That kind of resets everything on the client. All you have to do is pick higher ed, high school or higher ed. So this defines the level for each of the different products. And so, for example, biology is set as high school, and so on. Physical science as a high school or a middle school product. And so we have these different levels. So really what that does is it defines the content you're going to be using with the lab. All right. So so that's kind of like the client works. And so if you want to open a lab, then you just click on there and open, and that brings up a lab here. So like I told you, the level comes down here and specifies these worksheets. And so each worksheet has a corresponding, what we call a preset. It's kind of like having a great TA that comes in and sets up the lab for the students so it's ready to use. And so each worksheet has a corresponding title here that they would click. So if you assign Rutherford, then you would click here and click on this one, and that would take them to the lab with it set up, consistent with the worksheet that they would be doing. And then the step-by-step instructions would tell them what to do. So the metaphor or the motif that we've created here is like a real lab. If you have a real lab with a door, students go into a door, there could be a stock room full of chemicals, stock room full of equipment, and the room doesn't do anything, right? The room doesn't do anything until we as the teachers create the learning goals and the content and the procedures that they're going to follow and ask them the right assessments so that there's learning. So what we've done with these labs is we've just created the rooms. We've created each workbench as a standalone, completely open-ended environment where students can do anything that they would want and make any mistake that they could make in a real lab. And then, so nothing is baked into the lab to control the students. It's open. So what we do is we control the content to control the level and the learning goals that we want. And so this is an example of a lab that maybe isn't a lab replacement, because you wouldn't do Rutherford because it's dangerous and too expensive and too complicated, but in a virtual lab, we can do it. So here we have an optics table with alpha particle source, we have gold foil, and then we have this detector that we can detect the alpha particles with a phosphor screen. So nothing here tells them what to do, and so the procedures tell them what to do. So we may have them remove the gold and say, hey, what happened here? Oh, the spot got small. All right. What happens if I put this back? Then the spot gets big. What happens if I move my alpha particles over here? Well, now I don't get a spot, but I do get these deflections of alpha particles going through the gold. I can put it back in, and I see this spot, and we talk them through what that spot means. And then I can move my detector screen over here and see deflections of the alpha particles. I can move it over here and see it. And then I can go into persist mode and kind of save where those hit. And then we just guide them through with assessments how to interpret. So we just don't throw them into the deep end and say, okay, now what does this mean? We guide them and provide the structure to help students do Rutherford experiments. So for me as a teacher, you know, I don't like regurgitative questions that says, what did Rutherford's experiment teach us about the atom? When the student regurgitates, taught us that the atom was empty space, but they don't understand how he came to that conclusion. So for me, science is all about teaching students how they come to those conclusions, how to do experiments. And so we allow them to do these different kinds of experiments. And so I can move this back here, take it off persist mode. I can click here on the record button, save it. I have a lab book here. Now we can record the experiment that they saw here and put it in their lab book. And then they can save these lab books as files and keep them and reuse them and hand them in to teachers. So this is one lab. This is our quantum lab. Kind of showed you how it worked. This is the stock room for this lab. It's got lasers and electrons and super light bulbs, and students can mix and match and build anything, or the lab activities will guide them to do that. All of the labs have these clipboards that do other preset experiments like Millikan oil drop. So here's the Millikan oil drop experiment. Change the voltage, measure the charge of the electron, do two-slit diffraction of photons, change the slit spacing or change the color and see how that changes the diffraction, or change the intensity of the light coming in. And so just lots of different things that you can do here. So photo emission of CO2 gas. So I could spend a whole hour showing you all of these different labs, but this is kind of how it works. So each one of these up here represents one of these lab benches. So you have the ability to do these worksheets, but you can write your own worksheets or have students go in and explore. Let me show you a lab that does reproduce something you might do or would do in a laboratory for general chemistry. This is the inorganic qual. So we can grab a test tube, and in this test tube, we show a real picture of what's in it. And then you have these 26 water or cations that you could add any order, any combination. So here's cobalt, copper, I can add chromium. Here's a picture. Now we can do these lab manipulations. I can do a flame test. Show the green for copper, do a flame test with a cobalt filter. And I can decant and copy this test tube. I can heat it up. I can measure the pH of the solution. I can measure the pH of the vapor, smell it, and stir it. I can also come over here and add these reagents, any order, any combination. All right, so it's an unlimited amount of chemistry that you can do here. So we allow for all of this. So I can do like pH 4 or go to pH 10 and see a real picture. You see what happens to the cations. I can go to add sodium hydroxide, centrifuge that. Now I pushed it, now I can decant. And now I've separated my chromium from my cobalt and my copper. I can go add ammonia. I could smell that and say, hey, it smells like ammonia because I have a blue nose. I can centrifuge that and decant, and I'm left with my cobalt. Go to pH 10. I can go add carbonate right here, form a cobalt carbonate. And so we have all the permutations for all the mixtures and chemistry that can go on here. And students can save their work in their lab book. They can create unknowns, practice unknowns. They can use the clipboard to do basic unknowns, or come down here and do advanced unknowns. So we have a whole slew of activities that students can do here. So, I mean, I could show you all these other ones. Let me just open this one to show you. We have titration experiments, beakers and pipettes and pH meters, conductivity meters. We have a stockroom full of acids and bases, monoprotic, diprotic, triprotic. Again, you can do presets. So here's a preset, weak acid, strong base, titration. Go and do it. Save this data to the lab book. So I can save this data. I can save this graph. They can see, monitor the reaction. We have indicators. We have barometric pressure for the day so that when they weigh something, they can do buoyancy corrections. So lots of different things, calorimetry, gas properties as well. So before I leave chemistry, so this is kind of how they all work. This is a general chemistry lab, kind of focusing on what we typically teach in different levels of general chemistry. People also use this for PChem as well, classes for some of the advanced things that it does. So any questions?
I have a couple people asking about OChem.
All right. That's Heather's nice way of saying move on.
We can stay here if anybody else wants
It's
us
okay.
to show more in general chem, but
All right. Let's do
OChem and
organic.
bio people would like to see.
All right. So yeah, let's go to bio after this. So this is organic chemistry again. We have two benches here: synthesis and qualitative analysis. Same idea. Worksheets for different kinds of reactions, qualitative analysis. So let's go and do this one. So this is the synthesis lab. Looks a lot busier. This lab, you know, like I said, each of the labs have their own personalities based on the experiments that you're going to perform. This one tends to be more procedural. So the idea here is for a student to be given a target compound and then figure out how to make it and then prove that they made it. All right. So it's not quantitative. You're not weighing out stoichiometric amounts to do that. The focus we wanted here was on the decision-making behind what starting materials, what solvent, what reagents, what reaction conditions, how do you prove how long, and how do you analyze and prove what you've made. So, you know, we never claim to want to do a lab replacement. Obviously, in this time in our lives, lab replacement is an important element of these labs, but these labs really were built to be augmentations to real labs. Help them with pre-lab, help them with post-lab, help them with lab makeup, have them do more complicated things, pull out the structure and see if they've understood the principles behind what they did in the real lab. So in this lab here, what we do is we have all these different named reactions, and in this named reaction here, it defines a set of starting materials, okay? So just because it's a starting materials for Diels-Alder here, for example, doesn't mean that you have to do a Diels-Alder reaction. So I'm not an organic chemist, I'm a materials chemist. And so I don't know very many of these reactions, but I do know esterification. So if I pick esterification, what you see here these are the starting materials. So I created these starting materials on this bench. So here's solids, here's liquids are over here. Then what I can do is I can pick any 0, 1, or 2 of these starting materials, any order. So I can take, for example, this acid, and I can take this alcohol, and I can add it. I can pick a solvent here. I don't have to pick a solvent. I can pick one if I want. And so I'm not going to pick one. I drag this out to my stir plate. So I close my stockroom. This tells me what's in it. Now I can add any of these reagents to my mixture, any one of them. So like I said, just because we picked starting materials that are for esterification, you know, any of these reagents can be used or no reagent. So I'm going to pick my sulfuric acid. Now I need to build it. So I could start my reaction by clicking on my stir button here, but that will take longer. That's not the appropriate way of doing it. So I'm going to add my condenser. I can add ice or a heater. Obviously here I want the heater. I want to add nitrogen gas. If I don't add nitrogen gas, it's going to blow up. If I heated it too long, it's going to turn it into tar. If I put ice on it, it could take forever to react, or I may get different products. So what we've really done is really specified all of the different possible reactions and products and mixtures that you can make here. So I got my reaction set up. I start it. Now what I can do is I can do a TLC and measure. So here's my starting materials lane. Here's my product mixture lane. And so I'm already making product. So I can save that to my lab book. And I can come here and advance my time. So I'll advance 20 minutes. Then I'll come over here and say, how far have I gone? And there, wow, I've really made a lot of product. My spots are almost gone. And so I'm going to save that, say, okay, maybe advance another 10 minutes. And we'll go here. We'll see where we're at. So I have finished my reaction. Now, you're going to ask about kinetics. So we use the appropriate experimentally determined kinetics, so the rate constants and rate laws associated with each of these reactions, so that each of these reactions take a different amount of time. They're not linear, they're appropriate rate laws that we're using. And part of what the student can do is determine what these rate laws are by the time. So now we're done. We made our product. Now we're going to take the subfunnel and work it up. So here's my product mixture. Now I can pick one of these aqueous reagents, pick either one of them, and you can do counter current extraction. The appropriate one here is water. So I'll add water. It adds a diethyl ether layer to it. Now I can pick my aqueous layer or my organic and pull it off, and I can analyze it. So my aqueous layer is still here. I can pull it off if I want to, put it on the stir plate if I wanted to do some other work with it. So here I need to analyze it. All right, so we have all of our analytical tools here. So I can do proton NMR. So here's the proton NMR. I could zoom in on peaks, look at their splittings. Each of these numbers represent the numbered peaks over here, and it gives you the integrations. Save that to the lab book. I can change this if you're doing advanced organic. We can add the carbon-13 spectra. So these are all real spectra. We acquired these from Sigma. We can do a mass spec. Look at the splittings. Zoom in. Look at their masses. Save that. FTIR. So there's your FTIR experiments, and save that as well. So now that we're analyzed, now they have to go through and analyze, identify their peaks, and prove what they have. Here's the lab book. Here's where all that data is stored. And again, they can save these. So this is synthesis. So, you know, a very large variety of reactions that you can do, and we have the spectra for everything. Qualitative analysis. So in this one, the idea is to give students an unknown compound, and they have to do wet chemical tests and use their analytical techniques to determine what it is. All right. So we have different compounds with different functional groups. All right. And then they can pick one of these, and the structures you're seeing are knowns, so they can practice. So they can say, okay, this is what it is. Then see if they can figure it out on their own. And then I can click aldehydes. And so this gives them the knowns. And this one over here on the right is an unknown, and it's randomly assigned. So this is unknown number 210. And they can't just cycle through until they get one they like. They get the same unknown every time based on their computer. So I can add that unknown to my round bottom flask, pull this out to my cork ring, unknown number 10, give the boiling point, percent carbons, percent hydrogen. Now they can do these wet chemical tests. So take an aliquot, do say Jones oxidation, and we're going to show a real picture or video of that test. So this is obviously positive, but the time it takes is important. So that's why we show, you know, the speed of some of these tests so they can get the correct data out of it. So I can pause this right here and I can throw it away. And then I can come back here and do the same tests, the same NMR tests, and go try to determine what it is. So that's how you can give unknowns. We have worksheets that step them through this process. We also have another way of doing these unknowns where you can do these random ones. So I'll go back to aldehydes, and it puts an aldehyde here, but it's not numbered. So this obviously is a solid. I could come over here and do a melting point on my solid right here. And so mouse over, and it's 115 Celsius, and go do FTI. Whoop, that was the mass spec, and go do FTIR and do their spectra. Now they can go through and analyze what that is. The way they report this one is through the lab book. So in this one, they would come through here, click report, and then they would type in the name of their structure. So I don't know. And then click submit, and then it gives them the real name. Then what they would do is click save, and then they would save this to their, wherever they want, save it on their desktop, and then they could upload that to your LMS or email it or whatever form, and have you grade their unknowns that way. All right. Any questions, comments about organic?
Brian, the question is, what is the file format in which they're saved?
So it's a custom format that we used. So no other program can really open it. So the way it would work, if you want to read them, because of copyright agreements we had with Sigma, you know, we couldn't really make this globally available in some easy way. So we could only restrict it to opening inside of our own program. So there's the lab book. And so I could open it here and then show them what that report is. And that's a way that you would grade it. But, you know, this isn't the only way that you can report results. We do have these worksheets, but I know that lots of instructors, lots of professors would have students, you know, do screen captures of their spectra or screen captures of their results and put that into some type of Google Doc or Word file and upload that as a report. So that's certainly a viable option to do that. All right, biology. So same idea, worksheets, workbenches. So I do have to give a little background on what we are trying to do with biology. So I'm not a biologist, so my collaborators on this project were Keith Crandall at Washington University in DC, and Riley Nelson, who's at BYU. And the idea was to teach students modern biology. And so for them, modern biology is looking at and organizing and classifying species based on various ways of studying them. So we have here microscopy as a way of studying species, genetics as a way of studying species, molecular biology Ecology, and then we have a lab bench here called Systematics, where we then take data they may have collected or data that we give them for them to go through and classify species and understand how different species are related to each other, given different taxonomies. So just to give you a high-level view of some of these. So this is the microscopy lab. So we have these different microscopes, TEM, SEM, compound, dissecting, field scopes. And what you can do is we have a species selector that's in each of the labs. And so you can pick these different species. And then each of these species you can then look at on certain microscopes. So we have collections of images from these different microscopes that you can see. And so our database is about 3,500 images that mostly we took. And so, and then they can explore it. So and then the species selector allows you to organize these species in different ways. So animals and bacteria and protista and so on. So here's protista, I can click paramecium, and then we have like video of paramecium as they swim along. So here's another one, different scale, or we have different stainings that to expose different parts. And then you can look at it under a different microscope and say, wow, this is an SEM. So look at that magnification. So look at what a different picture it shows us based on the microscope that we use. So this is just a way of looking at and exploring different species. So for genetics, what we have is we have these different species here with different models. So I can pick fruit fly, for example, and you have all of these different traits, and you can pick multiple traits or one trait. So for example, I can do head shape, or I can pick and head shape eyeless and pick both of them. And then I can randomize their genotypes. So I want heterozygote here. So I'm going to push this button until I get something that I like, or you can specify it manually. And then you can specify a genotype for the parents and then do a cross. And then as you do the cross, it will show you the statistics of what you're going to see for the different offspring. So female, eyeless, leg-headed, female, eyeless, wild type, and so on. So here's the females, this is the males. So you can see each of the statistics and then analyze what's going on. So these aren't simple Mendelian genetics. We have all of the different genetic models. So, just to name a few, we have epistasis, we have incomplete dominance, we have sex-linked genes, and then what you can do is go through and see each of the different offspring and see, there you go, an eyeless with a leg grown out of the head, and so on. So students here, instead of just saying, here's data, do a Punnett square, collect the data and see what it means and see what the different gene linkages do. And so it's all based on real data. So this isn't idealized. This is the real data that's published in the literature. So here's molecular. So the idea here is what we did is we took all of our species and we created a mini genome. So we went to GenBank and extracted gene sequences for common genes amongst families of species. So some species may be 20 genes, some of them may be 80 genes. And we created this mini genome. And what you can do is take an epinorth tube, for example, and take a barnacle and take a DNA sample and add that. Now you can take your nucleotides and you can take your TAC and you can add that. And then what you can do is open, and then you have to pick the right primers for the right gene that you're trying to target. Obviously, this is the hard part. So the worksheets tell them what primers to pick, but you can add the right primers. I don't know what primer to pick. So I'm going to clear my lab and I'm going to go do a preset. So here's my preset for a fruit fly and a housefly for an ND1 gene. We want to see what's the same, what's different about that gene sequence. So I add it to my PCR and then close my lid, turn on my controls. I can control the temperature, duration, number of cycles, click run. Now I'm going to advance my time. We have a little animation that we can play here to show the different steps that's going on. So I'm just going to advance my time and get done here so I'm complete. Then what I can do is turn on my electrophoresis here, and I can take my pipette and take a sample and add it to one of the lanes. I'm going to add it over here to number 7, for example. And now I can change my duration, change my voltage, change my polarity of my electrodes. I'm going to click on, advance my time. So there's my tracking dyes. So I did a gel. Now it turned off after 10 minutes. Turn on my UV light. Use this to kind of measure the approximate mass of my samples. Now I can turn this off, and now I can actually do a sequence. So we have a sequencer, so I could come and add that, close it, and then I can click start and start advancing my time and start seeing the sequences as they come out, and I can show a chromatogram of these as they come out. Again, I'm going to do a preset here and just kind of show what the final answers are. So here's the sequence for, notice that they're pretty similar, but they're not exact. So these are the sequence. There's the fruit fly, there's the house fly, and then you can save these to the lab book and save this one to the lab book. Then you can open your lab book and analyze them, all right? Shift them. You can copy them into different formats and put them into web pages so you can analyze them that way. So ecology, this is pretty unique. We don't know of anyone else who has an ecology simulation. You can take any of these species and mix and match them, put them into different biomes, control your biomes, northern, southern hemisphere, latitude, elevation. So for example, I can change my elevation to 1,200 meters, or I can change my biome to, say, the tundra, and then change my elevation again. And then we show what the biome looks like, nutrient level, cloud cover, weather, what the weather is going to be, and then how does that affect the species, and how does it affect the species if they, kill each other. So clipboard, another preset, here's wolf and elk in a temperate deciduous forest. Click play, and then what we do is we can track their population, their biomass, their energy, save this data to a lab book, have them analyze it. So it's spring, they get births, populations decline. The wolf is actually increasing, the elk is decreasing because our initial populations were imbalanced. So we're going to see that over time, the elk population is going to crash, which means that the wolf will crash, and then they'll set up into a systematic, periodic up and down cycle for their populations. So systematics just let you go in, and what we do is we create a keep and discard pile here for species, and we have different schemes that you can use, different, like a Linnaean or a simple scheme, and let students go through and organize their species. We have glossaries. So for example, what if you want to say, where is my bacterium? So I say bacterium, and I'm going to mouse over eukarya and say, okay, here's the traits that I should see for eukarya, or this is what I should see for bacteria. And if you don't know what that is, you can click on that, and here's the definition of what that term is, and then they have to go through, and you click on that and you say, hey, it's still in my keep pile. So which one is it? Is it this one? That's wrong. So maybe I'll come back to this guy. So I'm right. So which one is it? So they can go through, and it's just finding how they organize themselves, and you can organize it this way and zoom out to see the schemes and different ways of organizing species. So that's biology. Any questions?
There's a call for physics, but let's see if anybody wants to chime in on physics or chemistry questions.
All right. So I know that we had one of you who were teaching 6th grade. So we do have a physical science product here with content that's geared towards middle school or high school. All right. So the physical science product, what it does is it brings all the lab benches for chemistry and physics together into one spot. And then we have content that you're going to want to use that's written for the middle school, or you can change the level to high school, whatever suits you best. So I just want to kind of show you that's an option to kind of put it all in one place with middle school or high school level worksheets. Physics, obviously, doesn't have all of the chemistry worksheets, lab benches in it. So in physics, what we do is we have these 4 physics-centric lab benches of circuits, optics, density, mechanics, but we also put in the 3 lab benches that we use in general chemistry for quantum gases and calorimetry, since those topics, particularly in higher ed, but also in high school to some extent, are also taught in a general physics course. So same idea. Here's worksheets. I think this is the higher ed worksheets. And we have these. So the major bench that you would use for physics is mechanics. So in the mechanics lab, we have this virtual 2D table that we can do experiments on with a camera that's looking at it. So I go into my stockroom, and in my stockroom, I have these different gravities, frictions, forces, like a rocket or a plunger, different objects that I can act on, and ramps in a solar system. So I can mix and match these different kinds of things to do different experiments. So for example, a simple experiment with a ball, and I'm going to have air friction, and maybe I want downward gravity, and I'm going to put a plunger. All right. So I have these objects. I click on my table. Now what I can do is I can put this ball anywhere I want, and now I can do an experiment. So this is really common for me. For what I like to do is I like to do this experiment. It's like, hey, put a ball out there, and you ask the students what they expect to happen, and I don't know, 80% of them are going to say, hey, that ball's going to drop. So if I click start and let the demons go, so the ball is going to do, nothing happens. Okay, time is going down here, but the ball doesn't move because there's no forces. So we can start talking about Newton's law. So I can reset that. Now I can go in there and put in some gravity. And now what's going to happen is it will fall. It will move in the direction of the gravity. But notice that my acceleration is constant, and my velocity is just going to keep on going up and up and up. So what if you want to change gravity? So you can click on parameters, come down here to gravity, and you can pick another planet's gravity, or you can type in the gravity you want or the number of G's. What if you want to change your object? You can change the size of your object to meters, or its mass, or where its mass is distributed. Is it a ring or is it uniform? And so you can always have complete control of your coordinate systems as well, and your units. And you can save data. So let's say that I want to go see what happens if I put in air friction. I can come over here in recording, click start, and record this. And note now that my acceleration is decreasing because I'm going to reach a terminal velocity, and they can go figure out what the terminal velocity is going to be. So I can click Um, I can pause, stops the recording, come to my lab book, and then here's all that data that they can copy all the data and then analyze it in in a Google sheet or Excel sheet and start graphing and plotting and then determining things. So, and then you can control what they can control what they save down here with the different check marks. So I can reset this, and then I can move my air friction off, and I can put a plunger on, and I can put the plunger any angle I want, click force, and show what that motion is and reset. So I'm going to return the items. So lots of things that you can do. Billiards, so I call this the bucket of balls. You can control your elasticity of collisions. You can do a 1D or a 2D. You can change your diameters. You can make the diameters and the mass the same or different for each of your balls. Change your friction, change what they're made of. So, so ramp motion, okay? You can do radial motion over here, like a radial gravity sink, and click on your rocket and set up and say, okay, hey, how do I go over here to my forces and change the duration and the force so I can get a circular orbit? Or what happens if I were to put in some air friction? So solar systems, you can do all of the solar system motion as well. So I'll show you like inner planets, click start, and track their positions. You can change it to be on the plane, or you can zoom in. So here's Mars. I'm going to zoom in on Mars, and I'm going to slow down to 1 day every 10 seconds. So there's the mass of them. There's their moons. I can enlarge them so I can see them a little bit better. I can zoom in on the planet and see the moon from that perspective. So that's what you can kind of do in mechanics. So it's most powerful to have students design their own experiments. We have presets, but design them, control their variables, and know how to do that. Circuits. So in circuits, we have an engineering paper and a breadboard, and they can take all these components, resistors, inductors, capacitors, light bulbs, and they can build a circuit here on the breadboard, and it'll reflect here on the engineering paper, or they can build it on the engineering paper and see what happens on the breadboard. We can do, here's a charging capacitor. So we have oscilloscopes, and we have function generators. So we can build up to 35 component circuits on that. Here's optics. So in optics, we have different kinds of objects and lasers. We have a virtual eye, lenses, mirrors, filters, prisms. So I'm just going to go do a preset here. So here's a concave mirror of an eye between the focal point. You can control here on this mirror, if it's a flat mirror, what the radius of curvature is, and then measure what this magnification is on this object. You can go do it at, past the center of curvature. You can do it. And so we have closer than the focal point. So all of these different kinds of presets and laser reflections. So this is what you can do with optics. And for the 6th grade teacher, this lab is pretty useful to do density and buoyancy. So we have all of these different materials and all of these different liquids. So I could, for example, pick my different liquids. I'm trying to go over here to, there's tar, here's soda, so I can fill my solution here. I can zoom in, make them measure what the volume is. I can take my beaker and tear it, then drag my graduated cylinder pour it in and have them measure the density of it. I can go put it back. I can measure the density of the liquid. Then they can pick, chocolate. So I'm going to go grab chocolate here. Let's go put this back and take my beaker off and re-tear it so I can come over here. Oh, I did cherry wood. There's cherry wood. There's the mass. Put it on top. Measure the volume before. Drop it. And this one floats. So let's go do another one. So we'll fill it. And let's go do copper. Put the copper on it and drop it. And now you can measure the volume difference by zooming in here. And then, of course, if they fill the wrong one, let's say they decide to go do sodium. I'm going to go drop my sodium in, and then it blows up. So lots of cool things you could do on the density lab. So we're at 12:55 for me, 2:55 on the East Coast. And any questions? I think that was a pretty good overview of all of the different labs, what they can do.
This is where you can chime in with any extra questions anyone has.
Let me talk about these worksheets a little bit. So these are PDF files. So this is a high school worksheet. So these are PDF files, and there's lots of different ways to, we have them all inside of our client, but you can download the ones you want and host them on your LMS. Students can fill them out by hand, take a picture, upload as a PDF. Sometimes some teachers just say, write out the answers and do screen captures of the relevant results from the experiment and put it into a report and hand in the report. You're certainly welcome to modify these. We can give you Word files. You can modify them to what you want to do. And you can, if you wanted to, port them into some type of HTML format for your LMS system. Really the guts of our product is the simulations, and the content here is just a means to get you started. And but feel free to do your own.
So Brian, Wendy was asking if there's an LTI to integrate, and I don't know how they get that. Somebody else was asking that earlier too. How do they get the LTI to be able to integrate it in?
So we don't have an LTI to integrate the worksheets into your LMS. In other words, the best way to do these PDFs is just put the ones you want and put them in as a download and as an assignment. That's what I do on my class at BYU. We do have an LTI integration for the licenses, all right? Mm-hmm. So what that means is that for some large online schools or other large schools, what we can do, normally what we do for most schools is we sell a code. So we'll make a code and it's good for 100 students, 50 students, 200 students. And we hand that code out, and students just put in the same code, and we just track how many of those get used. And when you get to the limit, then it stops working. But for some implementations, it's more convenient to not to have to hand out that code. And so we can integrate the code generation as an LTI into your system so that we can provide a link. The students click on that link when they click on, so the students sign in. So that link is hidden behind their login. They go to the course, they click the link. The link then queries our license system. It gives them a code, and then they put that code into their product. And each student gets their own unique code, and we can track. Then we can track who's using these codes a lot better. So that's a better way of code tracking. But it's up to you. We can provide the LTI and have your systems people integrated.
How long is the code good for, Brian?
So when they purchase a code, it's 1 year.
And they can use it, right, once they've got on their computer, they can use any of the labs they want. They can explore around chemistry, physics, bio, anything. So It's not just limited to one course.
Yeah, so you get the full suite and you get access to all the content. So If you're teaching an AP chemistry and a regular chemistry, then you may want to have the higher ed worksheets for one class, high school worksheets for the other.
Brian, show the new stuff real quick, just so they know.
Oh, that's a good idea. Thank you. So we are working on moving our content, our labs over to an HTML5. We hope to have our first one released, or maybe two released in the next month to 6 weeks. So this is what the titration experiment would look like. So it's the same underlying algorithms that we have before, same open-endedness, but we're changing the UI UX. So here, for example, is they have a beaker, and I can drag that beaker, drag it down here, put it under. I can go get my pipette, put it here. I can go take acetic acid, for example, and fill it. So now you can see the level filling in the beaker, and the card over here shows the level as well. And then I can just click here and fill that pipette. I can discard this if I want, go get another beaker, and add that to it. I could also, for example, take a beaker and add an indicator like thymal blue to this one. I can go add nitric acid to it, and I can come over here and put a pH meter in here, and so measure it. But then what I can also do is get another beaker and add ammonia, for example, and fill that. So you don't have to do a titration. You could do qualitative experiments with pH and chemistry. So you can drag that on top and pour from my beaker, and it goes through the different color changes as I go through. And then what I could do is drag this guy, put it on the stir plate, turn on my stirring, and pick one of my indicators, add that, pick my pH meter. And so you can see this card has the indicator, the pH. Maybe I want to get rid of these, clear them out, go get my burette, and maybe I want to titrate with sodium hydroxide, fill this up. So you see the card shows them more details. The image in the middle gives them a qualitative picture of what the experiment looks like. And then I can come over here and do my titration, and then see a graph of what that titration is. I can see they collect the data. And one of the neatest features that we're going to have is when you click here, you can save the state of this, creates a link that you can send to anybody else. They click that link, and by clicking on that link, it will regenerate the exact state that they have. So this is a way of showing the teachers where you were at, or sharing with collaborations with different students or things like that. So this is kind of how it works. Here's view the data. They can copy and paste that, analyze it. Same idea, same engine, but this is where we're going.
Brian, I'm not familiar with the coding side of this. What we have now here is HTML5, right? We're not in Java.
Right. Well, it's written in Java, but it's HTML5. It's Java based, let's put it that way, but it would be browser based, runs inside your browser. So we have other prototypes as well. Oops, that was the wrong site. Sorry about that. I want to go like, here's a prototype. We're changing the colors and everything, but this would be like the inorganic one. So here's cobalt, chromium, copper. You know, we're generating these images because this is designed to be lighter. It's for people with students with without heavy bandwidth, access to heavy bandwidth, or heavy access to really good machines. So same idea. All right. Sorry, for some reason you just saw all my emails. So any other questions?
Feel free to chime in if anyone has anything else they want to ask.
All right, I think we're good. Thanks a lot for all of your time. Thanks for coming. If you got questions, use our contact page. You can ask us or email us, and be glad to help you out. So thanks so much for your time, and hope to hear from you soon. Goodbye.